Good afternoon, everyone. And Dr. Quresh, this is not to be marked.
Yeah, I know.
I'm presenting some genetic thing in Fuchs dystrophy from Eastern India, especially from West Bengal. So you know that after 115 years, we still do not know what is Fuchs dystrophy. And in India, we see mostly late onset Fuchs dystrophy. This is sporadically familial. If you know that all EKs, FECD is the primary indication in US, about 80%, but the reports available from South India is about 10 to 15% of all EKs are for FECD. But in Bengal, the prevalence is much higher. And in our series, we have shown that it is 22 and 38% in BASAK and DMX series. So Tcf4 is the only gene on chromosome 18 which is extensively studied in various ethnic background. And this is cytosine thymidine guanine called Ctg18.1. If there is a repeat of more than 50, that is FECD manifestation, and maybe severity if it is more repeats. Western world, it is responsible, but less in India. No study from Bengal. The study questioned why we see more and is the genetic pattern is different. So that was the question. And we have collaborated with NIZED. This is in Odisha with government support. And this is what I am presenting. It is a part of the study we are doing for last 3-4 years. It is a prospective non-randomized case control study. 39 proved FECD patient who underwent DMEC and 59 available family members of the proband. These are called FECD subject, and 100 non-FECD individual at the blood age gender matched were taken. So there are different parts they have done. So we have counted the 50 repeats and more, and the pedigree and statistical analysis we have done. The sample size in both the arms are almost similar. And if you see that genetic association of Tcf4 Ctg, this is around 7%. Control is 2%. So it is not statistically significant. We have seen that there is incomplete penetrance with only in 10% of the family carrying this Ctg18.1 expansion. 4 of them showing FECD of characteristics, 3 asymptomatic, but if you see the family member, 80% almost, they lacked Ctg18.1 gene lacking. So these are their photographs, they have shown how they have studied. So this is the one of the individual family. If you see that, here there all the repeats in 3 members of the family. I have operated 1, 2 are waiting. The repeats are more than 100. And 2 other members, they have good enough that they have given blood sample from a 3-month-old child also. Highly motivated. But if you see from other states, there are few studies that are available. The Tcf4 is actually 17 to 34%, whereas if you see the Western world data, it is from say 81. And lastly, it is between 17 to 81. India, it was from Odisha, it was 34. From our study, it is 7 and 2, and it is not significant. Probably it is the lowest in the world, indicating other kind of genes and genetic mutation are responsible. So what we did more gene study in this, some of the family member, and we have seen that though the repeats are less than 50, there are FECD positive in this. So we studied the other LAMC and PGS1. They are positive for them. Like this is another family. They are also positive. And this is a huge family. You know that this 2 generation, 9 patient FECD, and I have operated already 3. 3 of them are waiting. Maybe another 2-3 years I will be operating them. So this family I am very interested to study more to go beyond this gene. So conclusion, unlike Caucasians and Western world ethnicity, this Tcf4 gene expression has little role in FECD genetics in Bengal. Probably it is multigenetic in origin in our population. Probably other epigenetic factors may have a major role. Very difficult to study them, long way to go. Thank you very much. Any question?
Thank you, Samar. You said that other genes could be implicated. So this is an ongoing study you're trying to find.
This is going for 3-4 years, so it will be going actually. So we are now studying. We initially took we didn't consider the spouses. So now we have we are going back to those because this blood is already in the repository, and we are taking the blood from the spouses, and we have seen that in some of the families there are some genes which are protective for FECD. So we have identified 3 families. So I have right now 59 families in 3 generations, but all of them, they are family members I am not getting. I'll go back, and I'm going myself with because I am deeply interested into it to get the blood sample
no,
at
least. I was
least.
just thinking that instead of comparing it with the West, which as you said is completely different from the, and Bengal is lowest even in India, why not go further east and compare with any literature from there, Myanmar, Thailand,
The Thailand there is there. They are like 44%, 49%, something
they are
like
also
this.
high.
They are high. And one interesting thing I always used to say earlier that Bengal, why is he more? Because we have been ruled by the British for 400 years. And other multi-ethnicity people ruled us, like German, French and Portuguese.
Yeah.
They all habitated like in 400 years. So I thought, and probably it is maybe that multigenicity is there. We do not know.
Okay.
Thank you.
Thank you for fascinating research.